Good morning, Ms. Johnson. I'm here to present to you a visual representation of the central dogma of biology. To start off, I believe that a little laughter is necessary, so I provided a joke. What did the DNA ligase say when it was confused at work? I don't understand. I was lagging behind. Ha 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 ha. Let's get started. Now, to start off, we need to understand what the process of central dogma is and what it does. In simple terms, the central dogma of biology explains the flow of genetic information from DNA to RNA to eventually proteins. As shown by the timeline here, the replication of DNA goes through transcription to RNA, translation to proteins. DNA contains the information that is needed to make proteins. The RNA carries the information to the ribosomes, and the results are proteins, which are functional. Now, to start off, we need to be on the same page with what DNA really is. DNA's scientific name is deoxyribonucleic acid, and is in the shape of a double helix, as shown right here. It is a backbone for all living organisms because it carries genetic instructions that make organisms different from each other. In DNA, there are two strands that wrap around each other to form the double helix, and each strand has a backbone that is made up of alternating sugar and phosphate groups. One of the four bases is attached to each sugar: adenine, cytosine, guanine, or thymine. These two strands are held together by hydrogen bonds, A with T and C with G. Since we know what a DNA molecule is now, we need to know how they replicate. Replication is a process in which the cell makes a complete copy of itself, as shown here. It allows for the continuation of hereditary information by allowing copies of the DNA to be transmitted from cell to cell. Keep in mind that replication occurs during the S phase of interphase. Now, replicating information is very complicated, so there are three main steps included: initiation, elongation, and termination. Initiation is when the enzyme helicase unwinds the two strands of DNA at the origin of replication, as represented by the helicase over here. You can basically think of this as a zipper of the DNA strands. This creates replication bubbles. Next, a group of enzymes called topoisomerase work to untangle the supercoiling of the DNA double helix ahead of the replication fork, which is represented by this here. It goes right there. After that, an enzyme known as DNA primase synthesizes short RNA sequences called primers. These primers serve as starting points for DNA synthesis, which is shown by this. Since primase produces RNA molecules, the enzyme is a type of RNA polymerase. Primase functions by synthesizing short RNA sequences that are complementary to a single-stranded piece of DNA, which serves as its template. Now, it is crucial that primers are synthesized by primase before DNA synthesis occurs, because the enzymes that synthesize DNA, which is called DNA polymerase, can only attach new DNA nucleotides to an existing strand of nucleotides. This brings us to the next step, which is elongation. Elongation requires the enzyme DNA polymerase. This enzyme performs the addition of new nucleotides to the new DNA strands and also acts as a proofreader to help prevent errors and/or mutations in construction from occurring. The building of the leading and lagging strand also occurs in this step, with DNA polymerase synthesizing new DNA strands in the 5' to 3' direction, which is the leading, and vice versa for lagging strands. So this is a leading strand right here, 5', 3', and this is vice versa, the lagging strand. So it would, so the lagging strand would be coming this way, reading from 5' to 3', and it would be vice versa on top of here. It is called a lagging strand because it is lagging behind the, because it is lagging behind, and then, yes. By this time, a new segment of the double-stranded parental DNA is unzipped. RNA primase synthesizes a new primer, and DNA polymerase 3 synthesizes a new Okazaki fragment in the 5' to 3' direction. An Okazaki fragment can be found right over here. Now you may be wondering, what is an Okazaki fragment? They are short sequences of DNA nucleotides that are synthesized discontinuously. This fragment is later covalently bonded by the enzyme DNA ligase, which is represented with right here, DNA ligase, and the Okazaki fragment. This concludes elongation. Now the last step, termination, occurs when two replication forks meet on the same stretch of DNA. These steps happen. Forks converge until all intervening DNA is unwound. Any remaining gaps are filled and ligated, and replication proteins are unloaded. This concludes the replication of DNA, and now it is time to move on to transcription, which happens in the nucleus. The process of DNA to be first used as a template for making messenger RNA is called transcription. During transcription, the message contained in the DNA molecule is rewritten in the form of an RNA molecule. Messenger RNA can then leave the nucleus and go to the ribosomes, where it functions as the disposable/portable copy of the protein building directions contained in the DNA. Just like replication, transcriptions happen in 3 steps: initiation, chain elongation, and termination. During initiation, a group of proteins known as transcription factors attaches to the promoter region found near the gene. Each gene or set of co-transcribed genes has its own promoter sequence. This sequence essentially serves as a signal that indicates the beginning of a gene. Next, an enzyme known as RNA polymerase attaches to the promoter. RNA polymerase is the main enzyme of the transcription. The RNA polymerase is represented here by the big circle. This would be the rewinding of the DNA coming back in. These are the template strands right here. That's the coding strand, and that is the unwinding of DNA as it keeps, the RNA polymerase keeps moving to the right. And you can see the RNA strand over here. RNA polymerase first separates or unzips the 2 strands of DNA. This only occurs in the area of the gene being transcribed. Next is elongation, where only one strand of the DNA acts as a template for RNA polymerase to direct the inclusion of bases in the newly formed RNA molecule. RNA polymerase reads the DNA template strand of one base or one nucleotide at a time and builds an RNA molecule out of complementary nucleotides. The new RNA molecule, known as the primary transcript, carries the same sequence as a non-template or coding strand of the DNA, except it has the nitrogenous base uracil instead of thymine. Now for the final step, termination. The RNA polymerase will eventually reach the end of the gene, and at that point, a stop or terminator sequence in the DNA signals the RNA polymerase to stop transcription and to release the RNA molecule. At this point, the RNA polymerase will unattach from the DNA, and now remaining portions of the DNA strands that are unzipped will reattach to each other. Now for the final part of the central dogma, translation. Translation is also taking place with 3 steps: initiation, elongation, and termination. So as over here, we can see that is still the process of termination, but it goes from mRNA to another mRNA, which contains a ribosome, which results in polypeptides. During translation, a cell's ribosomes read the information encoded in the mRNA molecule and use the information to generate/build a polypeptide. A polypeptide is a chain of amino acids. Each set of 3 mRNA nucleotides, referred to as a codon or triplet, codes for a specific amino acid. The 4 different types of nucleotides, A, U, C, and G, can be combined into 64 different codons. There are start and stop codons to tell the cell that the polypeptide is complete and that the translation should stop. This example carrying methionine, this example sequence will help with the understanding of translation as shown over here. So this can be found, the sequence over here just can be translated using the codon wheel and the rest. Initiation begins when the initiator tRNA carrying methionine attaches to the rRNA of the small ribosomal subunit. Together, they bind to the 5' end of the mRNA by recognizing the 5' cap. Then they walk along the mRNA in the 5' to 3' direction, stopping when they reach the start codon on the mRNA. During elongation, the sequence of nucleotides on the mRNA molecule is read in triplets/codons. Each codon encodes a specific amino acid, and then the correct amino acids are delivered to the correct place as specified by the codons on the mRNA molecule. So if we look at the example again, these would be the amino acids right here. These have been translated from up here. Amino acids are added to the growing polypeptide chain and increase the chain's length. Finally, termination occurs, which is the process in which translation is stopped. It happens when a stop codon on the mRNA enters the A site of the ribosome. The stop codon is recognized by proteins known as release factors. Thank you.